After studying this module, you will be able to know about the cell, nucleus and chromosomes, understand what is DNA and its role in living systems, learn how information is transferred from one generation to another, learn about genes, alleles and genome, understand what are dominant and recessive traits and their examples, differentiate between homozygous and heterozygous alleles, know what is phenotype and genotype, learn about monohybrid and dihybrid cross. Cell. A cell is the smallest structural and functional unit of life that is capable of functioning independently. Our body consists of millions of cells, not just the human body, but all living organisms from a tiny bacteria to gigantic blue whales are made of cells. These cells are capable of multiplying and forming more of their kind by dividing into two parts. Many cells together constitute tissues. Many tissues come together to constitute an organ, and many organs together to constitute a system. A cell produces energy by breaking down nutrients, uses this energy for metabolic processes, releases waste products, responds to environmental cues, grows and multiplies. To execute these varied functions, a cell contains many subunits that carry out specific functions. Some of these have been discussed in the following sections.
Nucleus. Let us start by learning about the nucleus. In cell biology, the nucleus, plural nuclei, from Latin nucleus or nucleus, meaning kernel, is a membrane enclosed organelle found in eukaryotic cells. It contains most of the cell's genetic material, organized as multiple long linear DNA molecules in complex with a large variety of proteins, such as histones, to form chromosomes. The genes within these chromosomes are the cell's nuclear genome. The function of the nucleus is to maintain the integrity of these genes and to control the activities of the cell by regulating gene expression. The nucleus is therefore the control center of the cell. The main structures making up the nucleus are the nuclear envelope, a double membrane that encloses the entire organelle and isolates its contents from the cellular cytoplasm, and the nuclear skeleton, which includes nuclear lamina, a network within the nucleus that adds mechanical support, much like the cytoskeleton, which supports the cell as a whole. Because the nuclear membrane is impermeable to large molecules, nuclear pores are required that regulate nuclear transport of molecules across the envelope. The pores cross both nuclear membranes, providing a channel through which larger molecules must be actively transported by carrier proteins, while allowing free movement of small molecules and ions. Movement of large molecules such as proteins and RNA through the pores is required for both gene expression and the maintenance of chromosomes. The interior of the nucleus does not contain any membrane bound subcompartments. Its contents are not uniform, and a number of subnuclear bodies exist made up of unique proteins, RNA molecules, and particular parts of the chromosomes. The best known of these is the nucleolus, which is mainly involved in the assembly of ribosomes. After being produced in the nucleolus, ribosomes are exported to the cytoplasm where they translate mRNA. Now we will study about chromosome. In the nucleus of each cell, the DNA molecule is packaged into thread-like structures called chromosomes. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. Chromosomes are not visible in the cell's nucleus, not even under a microscope, when the cell is not dividing. However, the DNA that makes up chromosomes becomes more tightly packed during cell division and is then visible under a microscope. Most of what researchers know about chromosomes was learned by observing chromosomes during cell division. Each chromosome has a constriction point called the centromere, which divides the chromosome into two sections or arms. The short arm of the chromosome is labeled the P arm. The long arm of the chromosome is labeled the Q arm. The location of the centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes. A human cell contains 23 pairs of chromosomes, that is, a total of 46 chromosomes. Out of these 23 pairs, 22 pairs are called autosomes, while one pair of chromosome is called as sex chromosomes, that is, X and Y. Males contain a X and a Y chromosome, while on the other hand, the females have XX chromosomes. Now, moving on to genes. Now comes the most important terminology called the gene, which is the foundation of genetics. A gene is the molecular unit of heredity of a living organism. The word is used extensively by the scientific community for stretches of deoxyribonucleic acids or DNA and ribonucleic acids or RNA that code for a polypeptide or for an RNA chain that has a function in the organism. Living beings depend on genes as they specify all proteins and functional RNA chains. Genes hold the information to build and maintain an organism's cells and pass genetic traits to offspring. All organisms have genes corresponding to various biological traits, some of which are instantly visible, such as eye color or number of limbs, and some of which are not, such as blood type, increased risk for specific diseases, or the thousands of basic biochemical processes that comprise life. The word gene was coined by Wilhelm Johansen in 1909 and is indirectly derived via pangene from the ancient Greek word genos, meaning race or offspring. The total complement of genes in an organism or cell is known as its genome, which may be stored on one or more chromosomes. The region of the chromosome at which a particular gene is located is called its locus. A chromosome consists of a single very long DNA helix on which thousands of genes are encoded. Now, moving on to DNA. Deoxyribonucleic acid is a molecule that encodes the genetic instructions used in the development and functioning of all known living organisms and many viruses. DNA is a nucleic acid alongside proteins and carbohydrates. Nucleic acids compose the three major macromolecules essential for all known forms of life. Most DNA molecules consist of two biopolymer strands coiled around each other to form a double helix. The two DNA strands are known as polynucleotides since they are composed of simpler units called nucleotides. Each nucleotide is composed of a nitrogen containing nucleobase, either guanine or G, adenine or A, thymine or T, or cytosine or C, as well as a monosaccharide sugar called deoxyribose and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar phosphate backbone. According to base pairing rules, A with T and C with G, hydrogen bonds bind the nitrogenous bases of the two separate polynucleotide strands to make double stranded DNA. DNA is well suited for biological information storage. The DNA backbone is resistant to cleavage, and both strands of the double stranded structure store the same biological information. Biological information is replicated as the two strands are separated. A significant portion of DNA, more than 98% for humans, is non coding, meaning that these sections do not serve as patterns for protein sequences. The two strands of DNA run in opposite directions to each other and are therefore anti-parallel. Attached to each sugar is one of four types of nucleobases, informally bases. It is the sequence of these four nucleobases along the backbone that encodes biological information. Under the genetic code, RNA strands are translated to specify the sequence of amino acids within proteins. These RNA strands are initially created using DNA strands as a template in a process called transcription. How does DNA encode and transfer information? Thus, DNA can be seen as a long sequence composed of the four base pairs A, T, G, and C. The information is essentially stored in the order of the sequence. It is similar to the manner in which alphabets of English come together in a particular sequence to form a word. This sequence is read by the cell machinery and converted into amino acids, which are the building blocks of proteins, and these proteins further form the building blocks of living systems. Let us understand this concept with the help of an example. Let us consider the DNA sequence G G T T T T G A T, the sequence of base pairs G G T wherever present in the chromosome, will code for the amino acid glycine, and TTT will code for the amino acid phenylalanine. This 9-unit sequence of DNA will thus be responsible for the formation of a peptide consisting of three amino acids: glycine, phenylalanine, and aspartic acid. Similarly, all the amino acids have corresponding three-letter base pairs called codons that signal the presence of a particular amino acid in a protein. These proteins are further responsible for carrying out all the functions in a living system. For example, the protein hemoglobin in the blood carries oxygen that we breathe to the tissues, and this oxygen breaks down nutrients, thus releasing energy. Sickle cell anemia is a hereditary disorder characterized by a deformed shape of red blood cells. It happens because the DNA of the affected individual codes for the amino acid valine instead of glutamic acid at a particular position in hemoglobin. Thus, a point change in the DNA sequence of an individual can have a huge impact on his or her health, and these changes can be transferred from one generation to another. Hargobind Khurana was an Indian American biochemist. He shared the Nobel Prize in Physiology or Medicine in 1968 for decoding that the order of nucleotides in a cell is responsible for the protein synthesis. Now moving on to allele and locus. An allele or allele is one of a number of alternative forms of the same gene or same genetic locus. Sometimes different alleles can result in different observable phenotypic traits, such as different pigmentation. However, most genetic variations result in little or no observable variation. Most multicellular organisms have two sets of chromosomes, that is, they are diploid. These chromosomes are referred to as homologous chromosomes. Diploid organisms have one copy of each gene and therefore one allele on each chromosome. If both alleles are the same, they and the organism are homozygous with respect to that gene. If the alleles are different, they and the organism are heterozygous with respect to that gene. The word allele is a short form of allelomorph or other form, which was used in the early days of genetics to describe variant forms of a gene detected as different phenotypes. Let us consider the gene responsible for color in flowers. This gene is present in both the homologous chromosomes, but while one is responsible for white color, the other will encode for proteins which gives purple color. These alternate forms of a gene are known as alleles. In genetics, a locus, plural loci, is the specific location of a gene, DNA sequence, or position on a chromosome. Each chromosome carries many genes. Humans' estimated haploid protein coding genes are 20,000 to 25,000 on the 23 different chromosomes. A variant of the similar DNA sequence located at a given locus is called an allele. The ordered list of loci known for a particular genome is called a genetic map. Gene mapping is the process of determining the locus for a particular biological trait. Diploid and polyploid cells whose chromosomes have the same allele of a given gene at the some locus are called homozygous with respect to that gene, while those that have different alleles of a given gene at a locus are called heterozygous with respect to that gene. Introduction to genetics. Genetics is the study of genes, heredity, and genetic variation in living organisms. It is generally considered a field of biology, but it intersects frequently with many of the life sciences and is strongly linked with the study of information systems. The father of genetics is Gregor Mendel, a late 19th century scientist and Augustinian friar. Mendel studied trait inheritance, patterns in the way traits were handed down from parents to offsprings. He observed that organisms or pea plants inherit traits by way of discrete units of inheritance. This term, still used today, is a somewhat ambiguous definition of what is referred to as a gene. Now we will study about the genetic terminologies. First is trait. It is a variant of a particular character which is passed only from parents to their progeny. If eye color is the character, then brown and blue eye colors are its traits. The most common genetic traits seen in human beings include the ability to roll tongue, albinism, hair color, shape of hairline, etc. A trait is always encoded by genes. A gene is a segment of DNA. Every human being is made of a basic unit of life called cell, and each cell contains chromosome which constitute DNA. Now, dominant and recessive traits. As we have already discussed, a sexually reproducing organism contains two genes or alleles for every trait. These alleles may be dominant or recessive. Recessive trait, a trait which is unable to express itself until and unless it is present in both the alleles, is called a recessive trait. In pea plants, white color in flowers denoted by small p is a recessive trait since it will be present only if both the alleles code for white color. Dominant trait, on the other hand, a dominant trait will be expressed even if its allele is present in a single copy. In pea plants, purple color in flowers, denoted by capital P, is a dominant trait. Now, moving on to homozygous. Bateson and Saunders coined this term in 1902 when both the alleles in the homologous chromosomes are the same, whether dominant or recessive, then an organism is said to be homozygous for that particular trait. In human beings, sickle cell anemia is a recessive trait. Heterozygous, if the homologous chromosomes contain two different alleles for a particular character, the organism is said to be heterozygous for that particular trait. Now moving on to phenotype and genotype. A phenotype, from Greek phenian meaning to show and typos meaning type, is the composite of an organism's observable characteristics or traits, such as its morphology, development, biochemical or physiological properties, phenology, behavior, and products of behavior, such as a bird's nest. Richard Dawkins in 1978 and then again in 1982 suggested that bird nests and other built structure such as caddisfly larvae cases and beaver dams are extended phenotypes. A phenotype results from the expression of an organism's genes as well as the influence of environmental factors and the interactions between the two. When two or more clearly different phenotypes exist in the same population of a species, the species is called polymorph. The genotype of an organism is the inherited instructions it carries within its genome. Not all organisms with the same genotype look or act the same way because appearance and behavior are modified by environmental and developmental conditions. Likewise, not all organisms that look alike necessarily have the same genotype. This genotype phenotype distinction was proposed by Wilhelm Johansen in 1911 to make clear the difference between an organism's heredity and what that heredity produces. The distinction is similar to that proposed by August Weismann, who distinguished between germ plasm or heredity and somatic cells of the body. The genotype phenotype distinction should not be confused with Francis Crick's central dogma of molecular biology, which is a statement about the directionality of molecular sequential information flowing from DNA to protein and not the reverse. Monohybrid cross, the mating between one pair of characters or cross made to study inheritance of single pair of gene is called monohybrid cross. We can depict the cross or mating in the form of a Punnett square. Suppose we wish to mate two pea plants which are identical for every trait except for seed color. Pea plant 1 has genotype YY and the color of seeds is green, and pea plant 2 has genotype capital Y and small y, while the color of seeds is yellow. The allele capital Y encoding for the yellow seed color is dominant. So the four quadrants in the Punnett square depict all the possible outcomes in the next generation. Thus, since the cross involves only one character, that is seed color, therefore it is a monohybrid cross. Next, we move on to dihybrid cross. Similarly, the cross between two pairs of characters or a cross made to study simultaneous inheritance of two pairs of genes is called dihybrid cross. Now moving on to what are generations. Now let us understand the important terminology involved in labeling generations. Capital P is the parent generation, where each parent is homozygous for the character under consideration. The heterozygous progeny resulting from this cross is called the F1 or the first filial generation. When this F1 generation is self-fertilized, it results in the second filial, that is, the F2 generation.
Let us now summarize what we have learnt in this module so far. Cell and its genetic components, the concept of genes, genetics and genome, structure and function of DNA, and how information is transmitted from one generation to another, the important terminologies in genetics.